Welcome to DNA Technologies Research Symposium.
Where we'll be discussing the ethical implications of each technology.
My name is Janavi Kolpekwar.
And I'm Durham. Now let's introduce our researchers.
I'm Gonzalez.
I'm Amshula Gajula.
I'm Veena Namboodiri.
And I'm Puneet Singh.
Every year, new sequencing technologies emerge which expand the scope of breakthroughs in fields like pharmacology, disease intervention, disease detection. Before we assess such modern breakthroughs, it's important to rewind on early DNA sequencing and techniques. So after the discovery of the double helix DNA structure during the early 1950s by Watson and Crick, came this idea of recombinant DNA technology. So we know, based on standard recombination processing, that we take our isolated gene from a gene donor, for example, and we ligate it with the plasmid, which consists of the restriction enzyme, and place it into a cell, right? Now, this idea of using restriction enzymes was introduced during the 1960s and 70s, and of course, biotech companies and pharmaceutical companies were very interested in this idea because using lab techniques, they can now, create a high production of restriction enzymes, so this idea became very reproducible. So this idea of recombination technology was very profitable, but it also kind of introduces the next topic, which is biotechnology. Okay, so following the age of recombinant technology began the age of biotechnology. So during the early 1970s, the biotech group called Genentech introduced this protein called the somatostatin, which produced the first transgenic bacterium. Transgenetics was the first take by Genentech, which was also later introduced during DNA fingerprinting by scientist Alec Jeffreys, where he utilized gel electrophoresis to distinguish between samples of genetic material. So the same way that restriction enzymes were used in recombinant technology, human DNA molecules treated with enzymes chop certain characteristics at certain points of the DNA, and this idea kind of are lateral. So later in the early 2000s, NIH, or the National Institute of Health, first approved gene therapy. At this time, human trials are still being ran for gene therapy. So now let's consider ethics. So some felt that these technologies kind of pushed the boundaries of science because it went against laws of nature, and it kind of went outside the parameters of religious beliefs. And many feared the term control, because it has potential to produce harmful pathogens, which could be harmful for maybe future generations. Next, Ethan will discuss the ethics of widespread gene sequencing technology.
Currently, access to new DNA sequencing technologies varies depending on a number of factors, such as complexity, cost, rules, and regulations. Generally, wealthier individuals will have higher access to these technologies, whereas individuals from lower socioeconomic backgrounds may have limited access. And this is due to the high cost of equipment, reagents, and the expertise that's required for DNA sequencing, as well as the need for specialized infrastructure and training. But that's the whole point of expanding these gene technologies, to make them more efficient, effective, and accessible. However, the widespread usage of DNA sequencing raises ethical concerns, including issues related to privacy, informed consent, and discrimination. For instance, widespread DNA sequencing could lead to the unintended disclosure of sensitive health information, which could jeopardize individuals in employment and insurance. Additionally, there's a risk of data breaches or misuse of genetic information by third parties. While widespread DNA sequencing has the potential to improve healthcare outcomes through the expansion of preventive healthcare and advance our understanding of genetics overall, it may also reinforce social and economic inequalities if it's not accessible to all individuals, regardless of status. According to a paper published in the Journal of Human Genetics, research suggests that disparities exist in access to clinical genetic services and in the efficacy of those services. African American women, for example, have been shown to have poorer access to the BRCA1 genetic testing than white women. Therefore, it's necessary that we establish appropriate regulations and oversight to ensure responsible and equitable usage of DNA sequencing so that we can all reap benefits entirely. These benefits include, once again, the preventive healthcare expansion, improved healthcare outcomes, and the advancement of our understanding in genetics.
Hi, I'm Veena, and I'll be talking about the target aid base editing technology. So what is base editing? So base editing takes a step further from the CRISPR-Cas9 process and any inefficiencies or limitations that it might have to actually do a site-specific, precise single base change. And so how it does that and the components that it uses is a CRISPR-Cas9 complex plus a base editing enzyme. And so looking at this diagram right here, as you can see, a guide RNA comes in and just kind of identifies the exact region. And then the Cas9 system can make a single break, which is important, not a double-stranded break. And from there, the base editing enzyme can actually make that base substitution. So it's able to perform C to T base changes or even G to A and vice versa. And again, without making any double-stranded breaks. So where is this used? It's used in gene therapy, it's used in precision animal breeding, and even to get the phenotypes that you really want and the outcomes that you really want. And so an example of this is a study that was recently conducted where they wanted to study the carotenoid accumulation in tomatoes, which is what gives it its bright and kind of ripe look, right, for sellers when they put it on the shelves. And so they took 12 tomatoes and they were able to make that change in 10 of them using this base editing technology where they saw a significant difference in the carotenoid accumulation by targeting those specific cluster of genes. And so what we do need to talk about though is the ethical implications of this. So it brings into question the safety, right? As this becomes even more prominent and applicable for humans as this technology progresses, right? And people discuss implementing it in germline therapy, right? For reproductive purposes. The safety of humans is called into question because of course, like this base editing technology doesn't work 100% of the time, and it can create problems where some cells hold that base edit and others don't, resulting in mosaicism and more. Another issue is with informed consent on the same topic, where we're looking at embryos, and I mean, at the end of the day, these embryos and the future generations aren't able to give that informed consent.
I'm Gonzalez, and I'm going to be talking about snapshot phenotyping. Snapshot phenotyping uses multiplex DNA typing to identify SNPs. More specifically, they identify their biogeographical ancestry to determine specific characteristics like how thick their hair is, how obese they are, and also their ear morphology. After 25-year-old Rhonda was found murdered, her autopsy ruled her death a homicide. Since CODIS failed to find the suspect matching the DNA found at the crime scene, FBI turned to snapshot phenotyping to see how the suspect looked. After 25-year-old Rhonda was found murdered, her autopsy ruled her death a homicide. With the help of the snapshot, Rhonda's family was able to gain closure after Ryan, her suspect, was finally put behind bars.
So now we have next generation sequencing. Next generation sequencing is a high throughput DNA sequencing technique that is able to very rapidly sequence through DNAs. For example, this has now cut the time to sequence one entire human genome from a few weeks to within the day. So in order to do this, there's a few intricate steps that we should take a look at. The first one is the library preparation. So anytime that you're trying to analyze any type of genomic DNA, you take that and you basically fragment or break it up into little smaller strings. And then you take a special adapter which you ligate onto either side. And the purpose of these ligators is that they have a complementary sequence on a flow cell, which the DNA will go and attach to. When they attach, the DNA will undergo bridge amplification, which is an extension of PCR, and they're able to create millions to billions of these strands in these clusters on the cell. After that, the reverse strand is cleaved because due to PCR, you'll have both the forward and reverse strand. The reverse strand is cleaved, and then you are left with just a single DNA strand, but millions to billions of those in the same direction. And that's really when the magic happens, because when we're ready to sequence, what we do is we create an environment where the polymerase is able to come. And this kind of visual shows that, excluding the polymerase. But basically, these nucleotides with the different colored fluorescent tags come in and they attach. And every time they attach, a light is emitted. And the emission tells us which color is attached, and therefore it tells us which nucleotide it corresponds to. Using this, we're able to get multiple single-stranded DNA, and this gives us several different strings, which we can then compare to a reference genome, which was determined by the Human Genoming Project, which took several years because they had to use slower methods like Sanger sequencing. And we're able to construct the actual human genome for which we were analyzing. So with this capability, the biggest ethical considerations are privacy and informed consent. Because we are able to so rapidly analyze so much human information for so many people, it's really of the essence that we keep this information private and make sure that only the people who have consented to this, only their information is used, and that this information that they have is private and it isn't leaked to any public or private corporations or entities.
Hi, I'm Amshula, and today I'll be presenting CRISPR prime editing technology. So first, CRISPR is where the CRISPR-Cas9 complex targets a precise location in the DNA and cuts the target DNA and inserts the new sequence to repair it to the preferred sequence. And this is a really good method, but there are some problems with it. The first one is the double-stranded break, which happens in the DNA, can cause future disruption in the genes when there are small insertions or deletions that may happen with DNA repair. Another problem with CRISPR is when homology-directed repair is used, there can be heavy dependent on donor DNA, and that's always not accessible. To propose a better idea, there is a new technology called CRISPR prime editing, and this uses the search and replace method. And essentially what that is instead of doing double-stranded breaks in DNA or heavily depending on donor DNA, it used a specified DNA site with engineered reverse transcriptase and directly guides the RNA to the specific spot and basically flips in the corrected sequence, so the edited DNA is encoded with precise. And some of the uses of CRISPR prime are in the sickle cell disease. So sickle cell disease is basically the AT to TA base pair mutation transversion and in the HBB gene. And what studies have shown is using CRISPR prime editing, there has been correction to wild type for up to 26 to 52% efficiency. And another disease that CRISPR prime editing has been used on is the Tay-Sachs disease. The Tay-Sachs disease is basically a insertion of 4 base pairs in the hexa gene. And in this, when CRISPR prime editing was used, there was a 33% efficiency rate for the hexa gene to be reverted back to wild type. Now some of the ethical considerations for this is, while there is a long way to go with CRISPR prime editing, there comes into question if we should correct mutations in the genome that cause diseases, and if so, who gets to decide who gets treatment and who doesn't. And if this does work, CRISPR prime editing could basically expand the scope and capabilities of genome editing and potentially correct up to 89% of known genetic variants associated with human diseases.
Thank you for attending our symposium today. We hope you learned something new.
Yeah, and as DNA technologies continue to grow every day, we hope that you can take away some of the ethical considerations.